So I called this presentation Bryophyte biology, but it is because it kind of covers the life cycle, and the nearest thing I could think of was biology, but it's not. Yeah, don't worry, it's not. It's not terribly boring. It's got a lot of detail, but it's also got a lot of pictures that show you what we're talking about. So the summary of part one might be worth doing at this point. There was a lot of information in part one. And you'll remember that we discussed about the fact that bryophytes are non-vascular plants that reproduce by spores, and that they belong to the kingdom of Plantae and are split into three phyla or divisions: the Mercantiophyta, which are the liverworts, the Anthoceratophyta, which are the hornworts, and the Bryophyta, which are the mosses. And we discussed the difference between not vascular and non-vascular plants, in that, for example, one of the main differences is the fact that the long-lived structures of a bryophyte, the leaves, the stem, and the rhizoids, all belong to the gametophyte generation, which is the haploid generation, whereas the diploid generation is the sporophyte generation, is much more short-lived and dependent on the gametophyte for nutrition. We also discussed about the fact that bryophytes don't have lignified vascular tissue, which connects to the leaves. And basically the way they cope with lack of water is to be desiccation tolerant. And they basically just shut down their metabolism when there is no water, and they restart it when water comes in the shape of either rain or flood or anything else. We also discussed about, we looked at different types of what constitutes bryophytes. So we looked at liverworts that can be thallus or leafy. We looked at mosses that can be divided into acrocarpous mosses, pleurocarpous mosses, and sphagnum. So the highlighted bits in yellow is just to show you this is what we're going to be talking about today. So looking at the actual life cycle of bryophytes in quite a lot of detail. And the aim of the session really is to learn about the bryophyte life cycle, talk a little bit about reproductive strategies. So we did discuss a bit about reproductive strategies last time, because we talked about how bryophytes can reproduce both sexually and asexually. And then we will look at the actual reproductive structures in the different types of bryophytes. The first slide is quite a lot to take in, so we'll look through it together. It basically shows the life cycle of bryophytes, and it splits, the black line splits between the gametophyte generation, which is below the black line, and the sporophyte generation, which is above the black line. And as you can see, the majority of what happens in the gametophyte generation, and the sporophyte generation just has the zygote, the embryo, and the capsule. But if we look at it from the beginning, in the capsule, which is the last bit of this sporophyte generation, spores are created by meiosis. They disperse, and they're called meiospores because they are created by meiosis. They disperse in the environment, they fall on the ground, and if they fall on suitable ground, they will then create the protonema. Now, this is an interesting point to look out for, because bryophytes are the only plant that has two stages in the gametophyte generation. So the protonema is the first stage. When you see this, basically what protonema is the very primitive stage of the life of a bryophyte. You wouldn't recognize it as a bryophyte if you saw it as at the protonema stage. It's either little sort of bits of effectively little threads of tissue that look a bit like an algal bloom on soil, for example, or little bits of thalli, like again little patches of tissue. And that's for mosses. So for liverworts and sphagnum, they're thalli, and for mosses, they are these little sticks of green tissue. So the protonema then sprouts, effectively buds, and each of the buds will form a leafy plant. And basically, the first part of the life of the gametophyte generation, the Bryophytes, is the protonema. And then once it sprouts and it forms, sprouts isn't probably the best way to say it, but once it creates a leafy plant, and then you have the sort of more long-lived part of the gametophyte generation, which is the gametophyte itself with the leaves, the stem, the rhizoids, and everything. The gametophyte will mature, will grow. When it's the right time for it, it will then produce the gametandria, what is called the gametandria, which are the bits that will produce the sperm and the egg. So the name for the gametandria in Bryophytes is archegonium for the female and antheridium for the male. So the antheridium will produce sperm and the archegonium will produce an egg. At this point, you have fertilization, if everything goes well and there's rain. And at fertilization, that's when you combine two sets of chromosomes and you then have the sporophyte generation, which is diploid. So the zygote starts and then the embryo will form a capsule, and the capsule will then start meiosis, create spores, and the whole cycle starts again. So that's sort of a brief run through of the whole cycle, and we'll now see what all this looks like in the flesh, in the plant itself. Starting from spores. Now, we don't often use spores in identification of bryophytes. So the only time you'll probably use spores is when you're trying to identify a genus of liverworts, which is called Fossambronia. And this is these are the spores of Fossambronia foveolata, which is basically Fossambronias look like tiny little lettuces, and I mean tiny, really tiny. They grow generally on a bare soil. And to be able to identify, take a good look at the lettuce and make sure that inside it, they have started to create a capsule. And you'll see it because it is either a green spherical structure or it's starting to go black. So it's starting to mature. And at that point, you can take a specimen, take it home, put it on your windowsill, wait for it to finish maturing. And when it's totally gone black, you can then open out the capsule and you'll get these lovely spores. And then from the shape of the spores and the ornamentation on the spores, you can identify the species. But just as an example, this is what a spore, Bryophyte spore looks like. And this is quite a large spore because I think that was only 400 magnification. So they're quite large spores. When the spores fall onto suitable substrate, we said they germinate and they will create these filaments, long filaments, if it's a moss. These filaments are called protonema, and protonema is effectively, I think it's Greek for first thread, because mosses create these tiny long threads. And from the threads, then you get these buds. They're very clear from the photo on the left-hand side. The green buds, they go on to produce a gametophyte, a full plant. We've said that only mosses create threads. Liverworts and sphagnum create thalli, which are like, effectively, when I Google to see photos, they are literally like mini thallus liverworts, tiny, tiny, tiny. And the problem with protonema is that most species, the protonema doesn't last any time. So once you start having full mature plants, you don't have the protonema anymore. It's dried out and gone. So there's only there's one species that I can think of that's quite common, where you can still see the protonema. With a bit of luck, that is something we will see on the face-to-face course. And if you look at the bank, which has this moss, which is called Pogonatum alloides, what you see the tiny little plants, and then you see this kind of greenish, I don't know, slime almost. And that is the protonema. But you only see it because this species has what's called a persistent protonema that stays there, even though the mature plants are already there. Most of the time, you won't see the protonema. So the protonema has created buds. The buds have created a gametophyte. So the gametophyte is the plant, what we see. It has stems, it has leaves, it has rhizoids, and it obviously looks very different, whether it's a moss, a liverwort, or a hornwort. They are completely different. Once the gametophyte has grown enough and has decided that it's time to reproduce, it will then produce Antheridia and Archegonia. So these are the gametangia that produce the gametes. So they are, of course, really tiny and really difficult to see, particularly the Archegonia. So the female bits are very difficult to see because they're often protected by perianth, which is basically modified leaves that envelop the Archegonia. So in the field, it's extremely hard to see. And the best picture I could get to show you what they actually look like was from a microscopic picture. So on the right-hand side, you have the Archegonia. They look like these long and sort of almost flask-like structures, which sort of bulge out a bit in the middle, and they contain the egg. Whereas on the left-hand side, there is these kind of sausage-like structures, effectively a long oval structure, and they contain the spermatocytes, which then produce sperm. And generally speaking, the shape of the Antheridia and Archegonia is more or less the same in the different species. It's more where they are located that varies between, say, mosses and liverworts, or whether the plant is monoicous or dioicous, and we'll talk about that as well. But there's knowing where they are and what they look like to be able to then find them under the microscope is an important issue, especially when you start getting a little bit more into more detail and try to identify slightly more complex species. Let's have another look at the detail of Antheridia and Archegonia, and also talk about this issue of monoicous and dioicous in bryophytes. Those of you who know about vascular plants probably have heard of monoecious and dioecious plants. So it's like holly, and you only get berries, so the female, the fruits, on female plants, and you get flowers that are male only on the other plants, on the male plants. So that is an example of a dioecious plant. Now, for the majority of vascular plants actually are not dioecious at all. They're monoecious. They have male and female flowers on the same plant. With bryophytes, it's slightly different, but sort of similar concept, and it's slightly different words. So it's dioicous and monoicous. But there are basically a lot of bryophytes that tend to have only female plants and only male plants. On the left-hand side, there is a moss, Prognathum alloides. This is the one that has persistent protonema. And if you look on the top left-hand corner of the picture to the left, there is a little bit of green slime, and that's the persistent protonema. But when you're looking at this moss, it is a dioicous moss, so it has only male and only female plants. The plants that have a bit of a red tip to them, they are male, and inside this kind of red cup, it's almost a cup of modified leaves. Inside the cup are the Antheridia. Female plants, you can see it a lot less, unless they have produced a sporophyte. So I've sort of circled at the top right-hand corner of the left-hand picture in red, a plant with what it looks like. Well, I know what it is because I know it's basically a seta. So it's the beginning of a sporophyte. And that tells me that that is a female plant. On the right-hand side, we have a liverwort, a thallus liverwort, which is monoicous. We know it's monoicous because it has got archegonia hidden under this flap of tissue, which is pointed at with a red arrow. So that flap of tissue underneath has a developing sporophyte, and before it had a developing sporophyte, it had the archegonia. The little kind of bumps on the same thallus further down the thallus point, which the yellow arrows point to, they are sort of little mountains, almost like little volcanoes, and inside them they have the antheridia. So because both antheridia and archegonia are on the same thallus, that plant is monoicous. And we've got a very clear example of how sometimes it can be a bit tricky until you know what to look out for to decide whether a bryophyte is monoicous or dioicous. But that's a start with a species which is very clear in this case anyway. Okay, so looking at Acocarpus mosses, there's a lot of Acocarpus mosses that seem to be the easiest way for you to sort of have a look at antheridia, because Acocarpus mosses tend to have their sporophytes at the tip of the shoot. They also tend to have the antheridia, if they're male plants, at the tip of the shoot. And in this case, both these mosses have antheridia splash cups. So the leaves are modified to form a bit of a sort of a cup, and inside the cup are the antheridia. When the sperm is released, when it rains, the splash cup will help sort of deflect the water and spray it out further, and that helps the sperm sort of swim further, hopefully, and find a female. The same is probably clear in the close-up of the Polytrichum pileferum. The red, really showy red cup has the antheridia inside, and again, if it's raining, you can imagine that it would sort of spray back all of hopefully the water with the sperm to go and look for a female. Much harder, so incidentally, it's much harder to see antheridia and archegonia in Pleurocarpus mosses, which is why I haven't got pictures of it, because they tend to arise from side branches, specialized side branches. We've got pictures of the sporophyte later, but just sort of seeing the specialized branch with only just the antheridia and the archegonia is extremely hard. So I've not been able to get any good pictures of that. Still remaining with Acocarpus mosses, not all Acocarpus mosses have the antheridia at the tip of the shoot. So even though generally speaking they do, there are exceptions as usual. And the genus Fissidens is one of these that has got basically, this is Fissidens bryoides, and it's clearly monoicous because it has at the tip of the shoot a seta, which would have had the sporophyte, which is out of shot. And then in the leaf axils, where the arrows are pointing, it has the antheridia held in a perigonium, which is basically, again, a modified leaf that holds the antheridia inside. And they are inside the pocket of the leaf pocket of Fissidens. So this tells us that this moss is monoicous, and once we know that we need to look out for antheridia in the leaf axil, together with a couple of other features, we know that we're looking at Fissidens bryoides. And the picture on the right sort of shows these little buds with the antheridia a bit clearer. So they are quite, they can be very different. Between different species can do very, very different things. when it comes to the location of the antividia and archegonium. I've been lucky to find these pictures of an antividia, again, quite nice, quite good magnification. On the left-hand side, you can see this structure that looks a bit like a sausage, and that's the antividium. But on the right-hand side is basically before the perigonium, which is all these modified leaves that hold the antividia, was split to, I think it was pierced to be able to find the antividia. This is what it looks like. It looks almost like a little bud, with all the leaves around. The leaves are quite hard. They protect it quite well until it's ready to get out and to release the sperm. So that's quite a nice picture of an antividium in Rhacomitrium affinis. So what happens, this is mostly what we've seen so far, Lacocarpus mosses. So what happens in other bryophytes? In sphagnum, antividia are generally held in the leaf axil at the tip of the branches, and very often these so-called antividia leaves, where the antividia are have a different color. So that in this case, this is sphagnum phallax, and it's got very orangey tips to the branches in the capitulum, which are highlighted, and basically the sort of orangey color leaves inside the leaf axil have the antividia. Now, in certain species of sphagnum, and especially in certain sections, the antividia leaves are red, bright red. And again, it's quite a good sort of diagnostic feature sometimes, because there will be species that have very red antividia leaves and species that don't. So sometimes it's all helping to contribute to the identification of a species. Now, talking about liverworts and weird and wonderful things. Liverworts always do weird and wonderful things. Some liverworts are like the ones that we saw before. They have a flap under which they develop a sporophyte hides, and then they have the little volcanoes with the antividia. But there are thallus liverworts that do something completely different. They actually have structures which have served the function of elevating the antividia to a slightly higher sort of altitude to allow for wider spreading of the sperm. And these structures are called antividiophores, and they're really visible in this species, which is Marcantia polymorpha. And they look like these sort of flat umbrellas, effectively, on the left-hand side. And they contain the antividia inside, and when it's time, they open out. Again, the water hits them, and they sort of fall from a slightly higher altitude, elevation, and that allows them to spread a bit further and hopefully find a female. Once the fertilization has occurred, so that the males, the antividiophores, develop first, and then fertilization occurs, and then these other weird structures on the right-hand side develop, and these are the archegoniophores. So instead of developing normal setae, these are sort of really quite tall structures and quite sturdy structures, and the capsules are inside the spokes of the umbrella. They are under there, and again they help dispersal of spores because they are slightly higher than the rest of the plant, and hopefully on a windy day they'll spread a bit further. But this is completely different from the other even thallus liverworts. There are a few species that have slightly more specialized structures for for reproduction. But this one is spectacular, and you have to see it because it's just so weird. So that's Marcantia doing its thing. In hornworts, the Antividia are contained in pits, Antividia pits. Don't forget, hornworts are much simpler plants. They are more primitive than liverworts and mosses. So they just have a sort of a container with the Antividia inside. When you look at this plant, this is a Phaeoceros laevis, and it does have so that if you look at the picture at the bottom, from the bottom of the picture and center and bottom of the picture, there are these thalli full of yellow, yellow dots. The yellow dots are the Antividial pits, and there are absolutely tens of them on one thallus. When you look at them in real life, you can just see a little bit of the thallus, and then you see all these yellow dots. So these are dioecious, and they only have dioecious, and they only have basically either male thalli or female thalli. The male thalli are completely covered with Antividia. So this is what they look like in these two species of hornworts. And if you look at the top of the picture, there is a female thallus with really young developing sporophytes at the top. Right, so moving on to the sporophyte. So we've seen the Antividia and the Archegonia. There's been fertilization. The sporophyte develops. And sporophytes are different in the way they develop between mosses and liverworts. So we're starting with the liverworts, which are slightly simpler as structures. The liverwort sporophyte has a seta and a capsule, just like a moss sporophyte, but the way they develop is slightly different. So first of all, the capsule will develop inside the perianth, and when it is mature, only when the capsule is mature will the seta then develop. And the seta is sort of see-through, and it's fairly thin and not very long-lived. So it will live a couple of days, day or two. It will just elevate the sporophyte, put it a bit higher, and the sporophyte is already mature, so it's ready to go. And when the time is right, the capsule has 4 sort of effectively weaknesses in the surface of the capsule that allow it to split into 4, or dehisce, which is the technical term. And it just basically doesn't have a lid, doesn't have anything. It just opens into 4 bits, and the spores fly out. One feature of liverworts, not just thallus liverworts, liverworts in general, is that they have an extra sort of help to disperse the spores. They're called elaters. So if you look at the picture in the middle, we've got our Fossambronia that we had before, and we've got the spores, but we also have these odd kind of spiral looking things. These are elongated cells that have a sort of reinforced kind of twisty part to the cell, which when it's enclosed in the capsule, is full of water. When the capsule opens, it starts to dry out, and when it dries out, effectively it starts to twist and deform, and it will be under tension. It becomes sort of under tension, a bit like a coiled spring. And at one point, it can't stand the tension anymore, and it will just flick off and return to its normal state and length. And in doing so, it sort of spreads the spores and throws the spores off, and it effectively helps dispersing the spores. Only liverworts have elaters. Mosses don't have them, but they are interesting little structures. Very nice to see under the microscope. So another look at the sporophyte for liverworts. These are leafy liverworts, and as I said before, the sporophyte tends to be enveloped in a modified leaf, which is called a perianth. So on the left-hand side, the really long, almost whitish or white-tipped structures on the left-hand side are the perianths of Cephalosia bicuspidata. They are really quite large compared to the size of the plant. And inside each of those, there will be a developing capsule. On the right-hand side, there is a radula complanata. This leafy liverwort has got pretty much rectangular perianths. And you can see from the picture that there are all capsules at different stages of maturity. So right in the middle, there's one that's just coming out of the perianth. It's still whole, and you can't see a seta, and it's just coming out of the perianth, and it still hasn't dehisced. Below, towards the bottom center of the picture, there is clearly a see-through seta and a dehisced capsule, for example. And then there's some perianths, which look sort of squarish or rectangular, that have sporophytes inside that are not, they're developing, they're not developed yet. So it's an interesting, another interesting sort of variation on the theme of what sporophytes look like in leafy liverworts. Right, moving on to mosses, and we've got Acrocarpus mosses. We said the sporophytes, generally speaking, arise from the tip of the sporophyte, of the shoot. On the right-hand side, we've got Polytrichum pileferum, which has clearly got its sporophyte just on its own. It's immature, so we've still got the calyptra on, the hat on, and the capsule isn't developed yet. So you remember that in liverworts, capsules mature first inside the perianth, and then the seta comes out. In mosses, it's the opposite. So the seta comes first, and then the capsule will mature, effectively, will ripen on the seta itself. So on the right-hand side, you have a seta which is well developed, and a capsule which is not developed yet. On the left-hand side, there's an interesting species, which is Plagiomnium undulatum, which has got a peculiarity. This species is able to produce more than one sporophytes at a time. Now, generally, most mosses and liverworts produce just one sporophyte at a time, because the plant itself has to nourish the sporophyte. It's quite a lot of work feeding one sporophyte, that I think you don't really want to put all your energy into feeding however many. This one's got 6 or something. It's quite a lot of work. So generally speaking, bryophytes have a hormone that will stop the development of further eggs once one has been fertilized. This particular species has the ability to switch off this particular hormone and produce lots of sporophytes. But we have to say that normally this happens if they're in a very settled, quiet, undisturbed environment, which has a lot of water around and a lot of way of getting nutrients. So only certain species from very stable environments will do this, of producing lots of sporophytes per plant. But it's interesting. It's an interesting thing. So I thought I'd show it, because it's just something that doesn't happen very often. We've already looked at this particular species, and we said it's a Dioicus species. But the interesting thing about this is that instead of having the capsule is very different from other bryophytes. Instead of having peristome teeth that open out and let the spores out, this particular species has a membrane, which is a whole membrane. It doesn't open out, but has little dots, little holes at the edge, a bit like a pepper pot. And it's the movement in the wind that allows the spores to come out of the holes on the sides of the capsule. So again, another peculiarity, if this is a very common moss, or very often on banks, on soil banks, and when it's got a sporophyte that's ready to release the spores, the white membrane really sticks out, and you really notice it as you're walking past, because it's got this kind of whitish look about it. And it's this pepper pot that will disperse all the spores through the holes. Another variation on the theme of sporophytes. This is the genus Andrea. So Andrea is a Nacrocarpus moss. It grows on rocks in mountains, very common on rocks in mountains. The difference with this one is it has got a capsule, which instead of splitting, having a lid, and the lid falling off, or having the membrane and the membrane having holes, the actual capsule itself splits into four. It's still closed at the top and bottom, but it has enough of a split for the spores to disperse. Another weird thing about this genus is instead of having a seta, as you can see, the capsules are held by this black sort of bit of stalk. That's not a seta, that's called a pseudopodium. And the weird thing about a pseudopodium is that it's not made of sporophyte tissue, it's made of gametophytic tissue. So it only has one set of chromosomes, and only the capsule is the sporophyte. So it's just another peculiarity which it shares with sphagnum. So sphagnum does the same thing when it comes to the pseudopodium. Right, moving on to the Pleurocarpus mosses. We said that Pleurocarpus mosses have their Antheridia and Archegonia held in specialized side branches. And in this case, you can clearly see the sporophyte coming off the main sort of plant in these kind of little short, stubby branches. And then this one has lots of sporophytes that come out of each of the branches. They are very difficult. Seeing the Antheridia is virtually impossible, but when they produce a sporophyte, then you can clearly see them. Here's another one, and in this case, again, you can see this little short, stubby branch coming up, and these seta are still immature, and with immature capsules inside. Moving on to sphagnum. Sphagnum is again a bit of a funny one. It has the capsule looks a little bit like a liverwort capsule because it's dark and it's round, but it's more complicated. It has a proper lid. You can see from the picture on the right hand side that it has a lid. And also it differs because instead of having gametophytic tissue that forms a seta, instead of having sporophyte tissue that forms a seta, it has a pseudopodium which is made of gametophytic tissue. So it does a bit of a mixture. It's a bit of a law unto itself. So some species of sphagnum produce lots and lots of capsules, and others you'll hardly ever see capsules on. It's just one of those things. And this is sphagnum sclerosum, which is very striking and does produce a lot of capsules. So looking at the sporophyte in hornworts. Hornworts, I should have put this at the beginning actually, because hornworts are very primitive and they're Sporophytes are also very simple, very primitive. There's no seta and capsule. There's only this capsule that grows from the bottom, and as it grows, it matures, and the tip will have mature spores inside, and it will basically just disintegrate and let the spores out, while the bottom keeps growing and it has immature spores inside, and they keep growing and they keep sort of maturing and so on. So very, very simple, nothing particularly fancy about hornworts. Talking about reproductive strategies, so we've discussed sexual reproduction, and at this point we have to say that not all bryophytes reproduce sexually. So we sort of touched on this last time, but it's at this point it's a good time to talk about that, because there are species that are known not to reproduce sexually at all and only reproduce vegetatively. Now, it's not clear whether it is because nobody's ever actually been to see a species, this particular species, at a time when it's produced a sporophyte, or whether it actually doesn't do it. But we can only sort of talk about what we know. So there are species that we are known not to reproduce at all sexually, or there are species that reproduce sexually in one continent but not in another, or species that in a particular area only have male or female populations, and they don't obviously reproduce sexually because they can't. So there are very odd things when it comes to reproduction of mosses and liverworts, and yeah, it is quite an interesting and liverworts in particular. There seem to be a lot of liverworts that don't seem to reproduce sexually, or that are not known in Britain to have reproduced, or that are known to only have male or female populations. So it seems to be a bit of a liverwort thing. So moving on to the sexual reproduction, and we can look at the various reproductive strategies for reproducing vegetatively. We've already looked last time at gemme, which is these basically discoid tissues, bits of tissue that the plant produces that are clones of the plant itself, held in these sort of what we call cups, gemme cups, that splash away when it rains. So very clever method of reproduction, and that's this particular liverwort, Thallus liverwort, Luminaria cruciata. Leafy liverworts also produce gemme, so these brown sort of gelatinous things on top of this scapania are clusters of gemme, much smaller, so not so much discoid, as kind of like almost like dust, which is held together in these sort of clots, groups of gemme, right on the tip of the shoot. Scapanias frequently produce gemme and different colors. So this one is Limeria, which produces chocolate brown gemme, but there are some that produce red gemme or green gemme. So it seems to be something that as well is useful to tell some species of Scapania apart. Mosses do it too. So this is a Acrocarpus moss, which is called Tetraphis pellucida, and it makes these lovely sort of modified leaves that form a cup with the gemme inside. They look like proper discoid gemme. One of the oddities about Tetraphis is that it produces the cups, the gemme cups, and it produces reproduces vegetatively, but at the same time it just goes hell for leather and produces at the same time sporophytes as well. So that it's not clear from the photo because they're out of focus, but on the left hand side there are sort of out of focus capsules, and you can clearly see seta as well. They're all from the same group of individuals that are effectively reproducing both vegetatively and sexually in for the same group of plants. So yeah, it's one of these plants that's kind of hedging its bets. It just makes sure that it will reproduce. But maybe it is because it grows on a slightly more unsettled substrate, because it grows on the sides of very old logs, very sort of decaying logs. So it probably thinks, if once this log's really decayed, one day it's just going to fall, and I've got to be ready to reproduce and go somewhere else. So maybe that's why it does it. Other methods of vegetative reproduction that is worth talking about is this deciduous branch tips. So some species, especially of Carpus mosses, you sort of feel the tip of the tips of the cushion they form cushions, and these bits of stuff come away, and they have a little bit of stem and a few leaves attached to them, and you can see them in the picture as sort of circled. They can just fall down, and again they will produce a new plant from that. The genus Campylopus seems to be really good at doing that, and also the Dicranum is another genus that does that. Deciduous branches, so these are again weird and wonderful methods of creating another plant that looks exactly like me. And this one has got quite a long stem and little rudimentary leaves. And these deciduous branches, this particular species produces hundreds of them. So this is Pseudotaxiphyllum elegans. You find it in sort of conifer plantation very often, or just generally the slightly dark woodland. And sometimes when you find it doing this, there's so many of these branchlets that you can't see the leaves, because they're all coming out of the leaf axils and they're obscuring the leaves underneath. Very striking way of reproducing. And another one, weird and wonderful way, bulbils. So these are mini stems with mini leaves, very rudimentary leaves. They normally, again, are in the leaf axils. This is Polia. Polia does it, and Bryum does it. They produce these little rudimentary bits of stem and leaves that will fall down and form a new plant. So I think we've probably seen, yes, we've seen a lot. As a summary, we can just remind ourselves, the bryophyte life cycle alternates between gametophyte generation, which is haploid, and the sporophyte generation, which is diploid. The dominant generation in bryophytes is the gametophyte generation, which is haploid, and the gametophyte generation has two different life forms. One is the protonema, and the other one is the gametophyte. Bryophyte spores germinate and produce protonemata, which are either small filaments in mosses or small thalli in liverworts and sphagnum. And this is the first life form of the haploid generation. The protonemata then produce buds, and the buds develop into mature gametophytes. Gametophytes produce the male and female gametangia, the antheridia and archegonia. Where and how the antheridia and archegonia are held on the gametophyte is different in mosses, liverworts, and hornworts. After fertilization occurs, a sporophyte is produced, and bryophyte sporophytes look very different in mosses, liverworts, and hornworts, and we've had a good look at that. And finally, bryophytes have different strategies for reproducing. One of these is reproducing asexually in very different ways, with gemme, with deciduous branch tips, deciduous modified branchlets, and bulbils.